friends, welcome back to my channel. I hope you are happy to see my videos. If there is any query, any requirements or any criticisms, then definitely you can text me and I will definitely try to improve my videos. So today's video is the continuation of our last video, that is cancer. In cancer, I told you about two important genes which regulate the cell cycle progression as well as the cell proliferation. And these two genes are, one is p53, another is the PRP, and both these genes are tumor suppressor gene. So today I will be talking about p53 gene, which is also known as guardian of the genome. So let's start. p53 is the tumor suppressor gene, which regulates the cell cycle progression as well as control the cell proliferation mechanism. Now it encodes a protein whose molecular weight is 53 kd. So that's why it is named as p53. And this p53 gene consists of 396 amino acids. And it is located within the P arm of 17th chromosome. That is, it is written as 17p13.1. That means within the P arm at 13.1 region, this p53 is located. Now, when one copy of this p53 is mutated, that leads to an very rare type of cancer which is known as Li-Fraumeni syndrome. And it mainly occurs when one copy of the p53 is mutated. But if both the copy of the p53 is mutated, as because it is a recessive mutation, then that leads to a normal cancer. And you may be surprised with the data that the 50% of the cancer that occur in our body may be due to the loss of this p53 gene. It doesn't mean that 50% of the cancer throughout the world occur due to p53, but the 50% chances of occurring cancer is due to the p53. So don't be confused. So now Li-Fraumeni syndrome occurs when the one copy of the p53 is mutated. But if both the copy is mutated, that leads to cancer. Now in normal condition, the amount of p53 is very low in the cell because it is readily degraded or destroyed by the proteasomal degradation mechanism, which is known as ubiquitinylation. That means this p53 is a phosphoprotein, and there is another phosphoprotein present in the cell which is known as MDM2. Its full form is mouse double minute 2. This is also a phosphoprotein, and in normal cell this p53 and MDM2 binds with each other, and MDM2 stimulates the ubiquitinylation, which is a one kind of proteasomal degradation mechanism. That means, say this is the p53 gene and this is MDM2. In normal cell, these two together form a complex, and that stimulates a special kind of mechanism which is known as ubiquitinylation, that is the degradation or the proteasomal degradation that leads to the cleavage or the destruction of these p53 genes, p53 proteins. So in normal cell the amount of p53 is very low, but when there is a DNA damage in the cells. When the cellular signaling system sends the DNA damage, then this p53 and MDM, both the proteins are phosphorylated. Now, in what way they are becoming activated, that I will say later on. But before that, we have to know why this p53 gene or the p53 protein is known as the guardian of the genome, because p53 not only regulate the cell cycle proliferation, but along with that it activates different cascade events that are somehow responsible for the cell care and the cell maintenance. So that's why it is known as guardian of the genome. So see, p53 activates the DNA repair system. That means when there is DNA damage, when the cellular system sends any DNA damage, so p53 gene is activated and it stimulates all those genes which are associated with the DNA repair system. So it activates the DNA repair system. Second thing, when there is any DNA damage, it arrests the cell at the G1 phase. So the G1S progression is arrested. It is due to the p53 gene. Third, when there is sufficient amount of DNA damage, huge amount of DNA damage, there is no way to repair those damages, then p53 force the cell into apoptosis mechanism. So another way, it promotes the apoptosis mechanism. That means it activates all those apoptotic genes which are responsible for the for activating the cell death mechanism. p53 gene is not only arrests the G1S cell cycle progression, but also it can repair the internal damages, as well as if it is not possible, then it forces the cell into the apoptosis for the removal or the death of those harmful cells. So due to this reason, the p53 is known as the guardian of the genome. Now, how these mechanisms are activated by p53, let's discuss. Now, in normal cell, p53 is present in a very low level, as because it combines with the MDM2 protein. But when there is a DNA damage, say for example, this is the DNA, the damage occur in this region. So when our body system recognize that DNA damage or sense that DNA damage, so it activates a specific kinase enzyme which is known as ATM. So it activates the ATM kinase. That activation of this ATM kinase leads to the phosphorylation of both the MDM2 as well as the p53 gene. So as a result, previously MDM2 and p53 in a complex form. But when the phosphorylation takes place, that leads to dissociation of MDM2 protein as well as the p53 genes, and the amount of p53 during this time also increases because the ATM kinase phosphorylates the p53 and the level of p53 also increases. And now this p53, it's a transcription factor, just I have told you. These activate a DNA repair gene. which is known as WAF1. It's a DNA repair gene. Once this p53 bind at the promoter region, that leads to the activation of the WAF1, and it produces a protein whose molecular weight is 21 kd. So that's why it is named as P21. So it produces a protein which is known as P21. Now this P21 protein combines with a CDK cycline complex that is actually located within the G1 phase of the cell cycle. This is called cycline D and this is called CDK4. So the CDK4 cycline D complex is responsible for passing the cell for G1 checkpoint to the S. So that is actually responsible for the cell cycle progression. Say for example, this is M, this is G1, this is S, this is G2. And now this CDK4 cycline is responsible for passing the cell from G1 to S checkpoint. So here it actually works. But when this P21 protein binds with this CDK4 cycline D complex, so that arrests the cell at these G1 checkpoints. Now this is P21. So cell is unable to enter into the G1S checkpoint because these CDK cycline complex activate or phosphorylates another protein, another important protein which is known as PRB. So this CDK4 cycline D complex actually responsible for the phosphorylation of the PRB gene and release the another factor which is known as E2F. But due to the CDK4 cycline D ability prevented by this P21, so it is not able to phosphorylate the PRB E2F complex. So as a result of that E2F is not able to synthesize any proteins which are responsible for passing the cell from G1 to S. Just I repeat again, the first of all, the ATM kinase is activated when there is a DNA damage. That ATM kinase is responsible for the phosphorylation of both the phosphoprotein, that is the p53 as well as MDM2, and that p53 activates the DNA repair gene which is known as WFA1, and it synthesizes or it encodes a protein which is known as P21 because its molecular weight is 21 kd. And now this P21 combined with the CDK4 cycline D complex. The CDK4 cycline D complex becomes inactivated and resulting into no phosphorylation of the PRB E2F complex. So E2F is not available, so no further transcription process or the protein synthesis takes place in the cell. So as a result, cell is unable to pass from G1 to S checkpoint. Now another function is P53 promotes the apoptotic mechanism. First of all, when there is sufficient DNA damage, there is no way or no chance to repair that damage or that cell, then the P53 do not activate or induce the DNA repair genes, that is the WAF1. It activates the another apoptotic gene which is known as BAX. So P53 activates Bax gene. It's an apoptotic gene. Now when there is DNA damage, p53 is automatically phosphorylated. So that's why the p53 remain available. So that p53 does not activate the DNA repair gene because the damage is much more. So it activates the Bax gene, which is an apoptotic gene. Now when the Bax is activated, it activates another anti-apoptotic or the repressor of the apoptosis gene, which is known as BCL2. It's a repressor for the apoptotic mechanism. So this Bax gene block the activity of the BCL2. Block. As a result, apoptotic mechanism is activated and the cell enter into the cell death process by the nuclear destruction, nucleus destruction, and ultimately through the phagocytosis, cell is eliminated from our body. So in this way, it can activate the apoptotic mechanism also. The another function of the p53 gene is the repression of the oncogenic activity. In our body, there are several cellular oncogene or the viral oncogenes whose activity is repressed by the RAS mediated pathway. That is called the repression of oncogene by the RAS protein. That is these RAS protein activate the another gene which is known as ARAF1. Now this ARF1 binds with the MDM2 protein and release the p53. Normally MDM and p53 is in a complex form and enter into the proteasomal degradation process. But when this ARF1 gene is activated, so it binds with the MDM2 protein, leading to this is MDM2 protein, and now it binds with the ARAF1. As a result, p53 is released and it represses all the oncogenic activation. So in this way, it not only protect our body but also maintain the genome. That's why it is known as guardian of the genome. But when this p53 is mutated, that means it carries the loss of function mutation. When both the copies of the p53 gene is mutated, say for example, both the copies of the p53 gene is mutated. As a result, the activation of the DNA repair gene is not possible. So there is no cell cycle arrest, that is G1S progression is automatically proceeds. Similarly, it is unable to activate any apoptotic mechanisms. So as a result of that, the cell becomes immortal. And third one, it does not activate the PRAF gene or the RAS mediated activity, so it is unable to repress the oncogenic repression also. So in this way, this p53, if this p53 is mutated, as a result, cell is unable to protect itself and it may be susceptible or prone to become a cancerous. So unable to repress oncogene, unable to check G1S progression, unable to restrict the cell, that is no apoptosis. So p53 is very important for regulating the cell cycle progression as well as cancer.